Now, the next skill is the question and answer session. We have a lot of policy, but I will look at least one of the first questions. So here I have the just completed the semester question for a one. First, you see, I start with just I understand the questions and then there's an idea of how the answers will look like. Yeah. They define the following medical outcomes. So, basically, explain that one of them is medical outcomes. So, how the church, so we have said in that, we are discussing about the bank that the definition of the outcomes. One of them is medical outcomes. So I would say that when you have some of the 1224 these there are applications in the field of medicines that are going to develop the athletes that are when you have sometimes the chemical reactions, the viscosity have a problem, one enzyme is not able to be produced, you have the particular or you have some specific changes in the chemical nature of the macromolecules with explanation that can be disease conditions. That is application for the health application, government application in medical situation. This is medical, but then we talked about what's it called, where we really mentioned the exposure about the system. So external chemicals, those external chemicals then it's a function of the chemical process. And when these happen, then these people has things that that's the system. While in biology, we talk about the physically how the enzymes influence the efficiency and the consequences result in the efficiency. So anything that has to do with the enzyme, talking about the enzymes themselves, talking about the functions of the enzymes, talking about if the enzymes are not there, the consequences, basically even how does the enzymes, how does the enzyme in the functions, is also covered. And then in molecular biology, we talk about the chemistry of nucleic acid and the chemistry of nucleic acid, information of DNA and how the DNA and the RNA cause the informations, or how the information in the DNA is being utilized versus RNA, how the RNA is being utilized with the protein. So all these things begin us to do molecular by technology. Basically, how is technology applied to this concept from the structural chemical process structure down to this process of conversion of information from DNA to DNA, which we call the application in the system, from DNA to RNA to installation, and from RNA to protein transformation. How does biotechnology or technology applied manipulate this seems some of those processes, like to design something of your interest in a particular conditions, this condition, without different field applications in agriculture, in industry to produce a particular protein, for example, insulin production of insulin using microorganisms, etc., etc. All these aspects of the concept of biotechnology. And then the next one is cell. It's a cell that is not developing functional unit. Life, you see, molecular unit of the bubble of this unit, said, alpha cellular is organelle. The organelle we have macromolecules. The supramolecules. The supramolecules we have macromolecules. From the macromolecules we have the singular molecules. From the singular molecules we have combinations of atoms of elements. And then the combination of elements we also called the electron talking to neutrons down to electron. Then the units question B says the relevance of medical biochemistry to our life sciences. And we just talked about how is the medicine, pharmacy, impact our so certain biochemical reactions and certain biochemical chemistry, how the identification of microorganisms, how certain microorganisms behave different from others. The presence of some genes that are found in some of these microorganisms make them to survive in certain conditions, make them to survive some certain food, make them to be able to degrade, for example, by falling at some ability to work. In the case of medicine, we just talked about the role of enzymes, some proteins involved in some certain disease conditions. Then we talked about the natural chemistry, how some of those molecules can be utilized to produce a drug. We talked about the enzyme inhibition by single molecules. Some of these new molecules, but those molecules that can be designed to produce a drug. This drug can inhibit some certain enzymes, but inhibit some certain interactions that can then use as a drug and treat a particular disease conditions, or even sometimes where impairment set during the biochemical process can lead to disease conditions we mentioned. So basically, how this linked to medicine, how this linked to pharmacy, how this linked to microbiology, all of this plus. Then the next question is number 2 says briefly explain chemical computer plasma. So we've already talked about the main constituent of plasma membrane, which we said that plasma membrane lipids and proteins. That the plasma membrane is the lipid and the proteins. The lipid definitions of different types of lipids, starting from the phospholipid, the main component, the phospholipid that forms the phospholipid bilayer. And then we have the cholesterol, this cholesterol, and how we maintain the rigidity of the membrane. Then we talked about the glycolipids, and glycolipids for cell matters. We also talked about the proteins where we structural, sorry, we talked about the integral membrane proteins and the integral membrane proteins. All these from the component of cell membrane. So we made a mention of a different type of phospholipids. Their structure, how does even the phospholipid comes together, what made them to be able to form the phospholipid bilayer. Remember, we talked about, since we talked about formations of micelles, we talked about formations of liposomes. So you have to just talk about these components. First of all, information that are basically made up of cell membranes and everything that are basically made up of lipids, the collection. The lipids comprises of the phospholipid, the material of phospholipid. I don't think you need to talk about this structure. Structurally, we just talked about the phospholipid that they have. It's polar hydrophilic and hydrophobic side. So hydrophilic and hydrophobic side, and we have the coming together of them to form a bilayer. Then you have the cholesterol, which is the rigidity of the cell membrane. And then you have the proteins, what's in structural tabular cell and the And the next question is, this one says the question between peripheral and integral. And I think the difference basically is on the basis of the association and the membrane. So you have the integral involved that interact extensively with the hydrocarbon chain of the membrane, where you have the protein spanning from the outside down to the inside, while in the case of peripheral, where it only interacts with some of the tonal amino acids that are positioned at the upper side of the membrane. Then the next one says differentiate between passive and active transport. So we say passive transport results in what the process of labor. So it's simple. It can be simple for facilitated targets involved the reading of molecules or the reading of higher concentration to the reading of low concentration without the most important thing here is the energy requirement. The energy requirement even though the movement from higher to lower is concentration driving, but a simple diffusion process for a facilitated diffusion. But in the case of active, this is where we have where energy is from the active water about the different energy is actually for gradient, a particular gradient is important. In three, you say briefly explain the importance of the glucose, which we discussed. In fact, I talked about that when we were discussing about the glucose, the structure of the glucose. So I took my time to talk about glucose and the importance of glucose. And I've already highlighted to you that I'm saying the question that about that. So please, you look at this information. I'll link out the discussion we had in the previous class. And the next one, say define if you know the terms monosaccharide, disaccharide, polysaccharide, for monosaccharide, we talked about the simplest sugar molecule that can be multiply further hydrolyzed. Then for the disaccharide, we say we have two sugars that are linked by glycosidic bond and we get with compounds. Therefore, the polysaccharide we have more than 2 up to 10 of them. For polysaccharide, we have many of them, many of those sugars. And then the have question also is how does the profile of the solution? Yes, I will discuss that one also while discussing what class where we say hydrogen ions are added to a buffer solution conjugate phase where the excess hydrogen ion acid, while the OH is added, the OH ion added, which is the base. They react with the acids present in the buffer to produce water and the conjugate phase, and this causes the electrolyzation and as such the substance water affected. It says these 3 values so we'll discuss about values please in our discussion. If the concentration of the solution is 4.2 times 10 raised to the power of -3, calculate the pH of the solution. It is also simple. Let's go back to the formula where the given pH is equal to negative log of hydrogen ion concentration. Simple, and this is the concentration of hydrogen ion is given. So just to substitute and then you have your result. And then say these say two properties of water, given different properties of water. Also, question 5 says briefly explain the whole level approach structure. Yeah, we talk about primary structure, secondary structure, primary structure, where we say it's divided by peptide bond formation. So we have amino acids linked with each other with peptide bond. This is basically primary structure. Secondary structure, we talk about hydrogen bond. The hydrogen bond leading to formation of alpha helical structure and look at the shift. And then we talk about tertiary structure, where we say we begin the formation of 3-dimensional due to additions of some other interactions in addition to hydrogen bonding. We have hydrophobic interaction, we have steric interactions, we have ionic interactions, we have sulfide disulfide also. And then unfortunately also we said this in addition to tertiary structure. So we have exactly as in tertiary structure, but in quaternary structure we have more than one for peptide. We will have two polypeptides and together, for example, for the tertiary structure, and then the tertiary structure also bring each other through the central photon, which include the hydrogen reaction, the hydrophobic interactions, the van der Waals interactions, and the ionic interactions, etc., etc. The next one says, did the cleavage site of the collaborators using the of the polypeptide, saying in the moment to see and plus 3 point. So you can, I think that you didn't have to have a lot of that where we have this acquirement of things and then they have the site where they create at the time. Question 6 says we define the members. Gives these biological functions of the discuss different functions like with examples. With example, with the classification of lipids here, we talk about simple lipid, we talk about complex lipid, we talk about dry lipids, and given examples also, all of this. So I think this is the end of the Wikipedia session, and the next one is in Wikipedia session. Then stop this and then